It's a bit difficult, and please bear with me. So what I'm going to do is try my best to describe to you the basic technology of CRISPR. So it's very basic, it's not really detailed, and I hope that everybody will try your best. You can search any other YouTube videos that is relevant, but one which is going to discuss is by using this link, yeah, this YouTube link, right? So first and foremost, before we can proceed to discuss what is CRISPR, you need to revise back a bit. You can just Google while we are discussing. So the structure of DNA, the structure of RNA. Okay. So what's the difference? DNA, we have double strand. RNA, we have single strand, and one of the nucleotide or one of the bases is different from DNA. So we have actually discussed that during our DNA-based biosensor. Remember when we learned about biomarker? Okay, ribose, deoxyribose is actually one of the structure involved in DNA and RNA, the sugar structure, right? All right. So next one, this is just fundamental first. You need to refresh. Of course, the ATCG, you still remember the bases, the nucleotide. What we will discuss, probably a new term, is what is palindromic repeats. Okay. Uh, and we will talk about virus and bacteria. Virus can have double strand DNA or RNA. Okay. So virus can also have DNA or it may be RNA virus. Just like COVID, COVID is RNA virus. Dengue also is RNA virus. Okay. It cannot have both at the same time, but it might have DNA or RNA. So this one we have discussed the complementary of DNA, and we also know that in the RNA, one of the nucleotide uh, is uracil instead of thymine. Yeah? So DNA has ATCG, whereas in RNA and single strand, it has uracil nucleotide instead of T. Yeah. All right. So you don't have to answer this. Don't worry. But this is just to refresh what we have discussed during the biomarker discussion. Okay. So why we are looking at CRISPR technology, it is because it is now quite a trendy biomarker. So we have discussed biomarker like DNA, antibody, aptamer, okay? Those we have discussed. So now we are discussing another cutting edge biomarker called CRISPR technology. So CRISPR is actually a short form. Yeah, it's not really the name is CRISPR like DNA antibody, but it's a short form of a quite long series of words which we will discuss. Yeah. So make sure if you don't know this, just a quick search, find what is a DNA, RNA, and then just refresh back what is ATCG. Okay. So this should be the first that you need to look at. So these informations can be downloaded from Putra Blast. Yeah. So now let's look through. So what is palindromes? Palindromic. So genetic palindromes are similar to verbal. Okay. It's the you can see that the complementary sequence, the 5' N to 3' N base pair sequence is identical in both strands. Okay, so you see CG, and then you come back to CG. Okay, so that is palindromes. Yeah. So this one, as I said just now, that I also still in learning process. Okay, this is a lot of molecular work, so please bear with me. And probably certain words that you have to know is about transcription. Yeah, transcribe. Okay, so this is a very common process that there are certain the process of protein. Okay, the process of protein formation. It starts with DNA, and then it is transcribed into RNA, the messenger RNA that initiate the process of protein formation. Okay, protein in our body. So this is basically the process involved. Yeah, start with DNA, transcribe into RNA, and then the messenger RNA will start to process the protein, any protein that in our body. Okay, so the word that you should be searching for is transcription. Okay, because we are going to discuss transcription. The series of how protein is formed. Okay, there's a lot of detail in here. Okay, but I'm not going to discuss how protein is formed here. You can just Google if you wish to know how protein is formed from messenger RNA, and before that is from the transcription of DNA to messenger RNA. Okay, so all these are happening in our body, and it's a very complicated process, but it's beautiful. This is what we are. Okay, that is currently happening in our body. Our hair is growing. We have to cut our fingernails, and all those enzymes or proteins that we are using in our body is through this process. Yeah, so it's good to know what's happening in our body. Okay, right. Chemical structure, I think this one we have discussed already in the biomarker section. So this one is done, yeah? Okay, right. So let's move to, we already record this, okay. The second slide, the second document, okay. Let's look at the second document, which is here, okay. Right, and this is the same document, sorry. Stop share this one. This is okay. Sorry, again. Okay. So that will be the prior knowledge that you have to revise back in order to understand the next one. Why is it not sharing? Just a moment, I'll try to work it out. Hopefully it works. No, it doesn't work. Just a moment now. Okay. Okay. Now you are seeing another piece of document. It's all in Putra Blast, as I mentioned just now. Please do download. Okay. So you need to put this um, document together during your 8 to 9 uh, listening to the recorded video, because from that video you have to find out the terms like what is protospacer, what is protospacer adjacent motif, which is called PAM, okay, and then what is guide RNA, what is Cas enzyme. There are many Cas9, Cas13. There's a lot of Cas enzyme. And what is single guided RNA? What is guide RNA already been mentioned. And then if it is possible, look for knockout gene, knock-in gene, okay? It is additional. And then we have all assignment. Okay, this one is not assignment for you, but this is by the invited speaker that we invite before this. Okay, and then so please notice all these terms. Okay, get to know all these terms. Okay, while watching the video. Okay, all right. So there is different type of CAS. So please be aware of what is CAS. Okay, CAS is the enzyme, and there are Cas9, Cas12, and Cas13. I think as long as we know the function is good, but I also don't know the difference between 9, 12, and 13. Okay, so make sure you download everything here. So now I stop share this one. Okay, just give me a moment. I need to take water. So, what is actually CRISPR? Okay, so the long name of CRISPR is, where is it? Yeah, it's clustered regularly interspaced short palindromic repeats. So, repeats here represent something. Palindromic is what just now that the backward and the forward has the same. Okay, so let's look at a few terms here. Protospacer, spacer, protospacer, adjacent motif, or pen. Okay, but before that, let's learn the history. Okay, so CRISPR stands for this. That's how we get CRISPR. And the mechanism of CRISPR-Cas, the most important from this technology. And then this is the main people are talking about, the gene editing. You can do gene treatment. If you don't want to get fat, remove the fat gene. If you are like possibility, your family has this heart issues. Well, people are talking about removing this gene related to heart disease, diabetic disease. So these are going on, and this is why CRISPR is, trendy. Talking about to use CRISPR in treatment of disease and treatment of especially gene-related disease. Yeah. And in biosensor as well, CRISPR has been used as the biomarker. Okay, so that's why we are discussing CRISPR in this subject. So look at CRISPR technology history. So in 1987, Japanese researcher has discovered, means observed, there is short direct repeats interspaced with short sequences. Okay, what does it mean is we have to move downward a bit. Okay, so what does it mean is this is the bacteria. Okay, this is virus. This is bacteria, Streptococcus bacteria. Okay, so what the Japanese researcher observed, there is a repeating gene sequence. There is a repeating gene sequence between the gene, the sequence of the bacteria. Okay, so what is that? Is sort of uh, a mystery, but then they know that it was observed that this spacer sequence, the repeats in the gene of the bacteria, gene sequence of the bacteria, there is a certain repeat, the same set of gene sequence repeat in the bacteria is actually from virus that is infecting the bacteria. So if you see the picture just now, um, so bacteria is the whole thing because bacteria is bigger than virus. So what happened is if the bacteria is infected with the virus, okay, there is some part of the gene sequence of the virus is copied into the gene sequence of the bacteria. And why it is there? Because the bacteria would like to remember the virus. The bacteria would like to remember the virus. So in future, okay, if the virus attack the bacteria again, it will recognize the genes of the virus, and it has this Cas enzyme, okay, Cas enzyme that can do or cut, yeah, this gene sequence of the virus, and when it is cut, it's sort of destroyed. You can see this is the cutting mechanism. Reinfection by the same virus and the Cas system. Okay, we will discuss this a bit more. It will cut, yeah, and once it is cut, and because virus is defined by its RNA or DNA, so once it is cut, now it's no longer function as a virus. Okay, so that's how the bacteria uh, defends itself for future reinfection. Okay, so for us humans, we more on antibody, right? Our antibody system recognize the infection, and we have our T cell, white blood cell attacking the infection or the virus infection. So so for bacteria, this is their defense mechanism. So that was the history of CRISPR, but of course, uh, a lot of works has been done. Okay, first to demonstrate experimentally the CRISPR-Cas9 mechanism. So it is the first two is more on observation, and the third one is experiment, and then the latest one that Jennifer Duna and Emmanuel Carpentier. So these ladies, okay, Jennifer from Berkeley University, and Emmanuel, she's a French, but she works with a lot of university. and they have won this Nobel Prize on their work on modifying the CRISPR technology. Okay, so and this is Zhang from Broad Institute. So Broad Institute is a research institute combining expert from MIT, from Harvard, okay, and they do a lot of CRISPR related technology. So there is actually a dispute between Zhang and Jennifer Doudna, who actually has the patent, uh, you know, who first patented the one that they found. So I don't know whether the dispute is still on or it's not. It is settled? I don't know. Okay. All right. So now, while discussing this, I will have to open the YouTube as well. Okay. So that we can have more understanding. Right. So this is the Nobel Prize. So CRISPR repeats. Okay, let's move on to the which one? Okay, so let's stop until yeah. So this is happening a few years ago. That this Chinese scientist has used CRISPR to have the first gene-edited babies. So he had impregnated a woman synthetically with embryos that had been modified to make them resistant to HIV infection. But since what he did is considered non-ethical, okay, so he was like, I don't know, he has to be prison for how many years? I don't know. I forgot. But I do read about his story, and he was in prison for how many years? I don't know. Okay, so it's not ethical to do that. Probably in future, I don't know. Okay, so let's look at, okay, I hope that you're seeing this. Okay, so I'm going to discuss while using this video of Dr. Matt. Huh? Okay, I hope you can hear this clearly.
Cas9. And it all began the same year I was born, 1987. Researchers in Osaka, Japan identified in the bacterial genome of E. coli these clustered repeating segments of DNA that tended to be short, but also palindromic, which meant...
Okay, clustered repeats of DNA. Clustered repeats of DNA in bacteria. It is short, but you can see it's palindromic. What does it mean? It has the reverse and the opposite, the forward and reverse side the same. Yeah. So let's look at.
They were read forwards the same way they were read backwards.
That is
Now, they also
palindromic.
found that these repeating palindromic segments of DNA were interspersed by spaces of DNA that seemed to be...
Okay, so these repeats were interspaced by DNA. So remember about the spacer, the repeats, the word spacer, the word repeats. Please take notes.
like random. Now, 13 years later, other researchers identified these exact same clustered repeating palindromic sequences in other bacteria and even archaea. And today we know that they're present in up to 50% of bacteria.
So what is the difference between bacteria and archaea? Okay, it's not archaea, it's archaea. Okay, so archaea has right. Actually, you can search. Archaea has this have cell walls without peptidoglycan, while bacterial cell walls contain peptidoglycan. So it's quite similar, archaea. Okay.
Bacteria and 90% of archaea. But back then we had no idea what these repeating segments and even the spaces between did. It took nearly 20 years from that original discovery to identify the fact that it wasn't these repeating segments that were interesting, but the spaces were interesting.
Okay.
Now, what we
So remember the word spacer and repeat.
called this entire stretch of the repeats and the spaces were CRISPR. And CRISPR stands for clustered, regularly interspersed, short palindromic repeats. What these spaces were actually fragments of viral DNA.
Okay, remember that I mentioned just now, when the virus infect the bacteria, so there's certain range, there's certain sequence of gene that is copied into the bacteria DNA sequence. Okay, so that small copied, it is called spacer. Yeah.
Just discovered that bacteria had a mechanism to be able to cut fragments of DNA from viruses that were infecting them and incorporate those fragments into their own genome as a memory to remember that invading virus. Effectively, what researchers had discovered was the bacterial version of the adaptive immune system. Let's now take a look at how this CRISPR system works and how we can utilize it for gene therapy. When a virus, like this bacteriophage here, attacks or invades a bacteria, injecting its viral DNA, the bacteria or archaea has evolved a mechanism to be able to identify the viral DNA, chop up certain sequences, and incorporate it into its own genome.
Okay, so chop a certain sequence and copy it into the bacteria genome, and it is called spacer, the blue one. Yeah.
As a memory of that virus. So an adaptive immune response. How does this work? So firstly, once this viral DNA is injected, the bacteria or archaea has this group of proteins or enzymes called Cas1 and Cas2. So here we've got Cas1 and here we've got Cas2. What they do is they recognize a particular fragment of this viral DNA and they chop it. So it's going to cut it there and it's going to cut it there. What it cuts, this segment here, we call a protospacer.
Okay, now you know the word protospacer. Yeah, remember the list that I have asked you to look into the terms. So the protospacer is the specific fragment of DNA from the virus that is being cut by the Cas enzyme. Cas enzyme is used to cut the nucleotide. Okay, so this portion specifically cut, it is called protospacer. Yeah.
We call it a proto spacer. Now your question might be, where does it chop? Does it just chop randomly or at specific areas? It does chop at specific areas. It will identify a sequence or motif of DNA on this virus that we call a
Okay, it is called PAM.
protospacer adjacent motif, also known as a PAM.
Okay.
And once it identifies,
so remember that protospacer adjacent motif, PAM.
This PAM region, it will cut a few base pairs upstream of it.
Okay, so there are certain like 3 or 4 base pairs. It is cut here. Okay, so it's the genes that is interested that will become the spacer is here, but it cuts 2 or 3 or 4 before the protospacer, and this is how they identify. Means the Cas enzyme identify where to cut based on this PAM. Okay.
So it's now created or cut this protospacer, and it takes this protospacer and incorporates it into the bacterial genome. Once it's incorporated into the bacterial genome, it is now called a spacer. So here we have a spacer from a virus. We've also got another spacer here, which is from another virus. Now, the areas in between the spaces, these are going to be the clustered palindromic repeats, that when a spacer is placed in, a clustered palindromic repeat is placed afterwards. So I'm going to call these repeats.
Okay, so remember, repeats is from the original bacteria genome, right?
Repeat and repeat. What we call this entire collection of DNA, where we've got various repeats and we've got various spaces, we call this a Crispr array. So this whole thing here is a Crispr array. And so if we break that term CRISPR up, right, clustered, regularly interspaced short palindromic repeats. Clustered because it's in a particular grouping or area of DNA, regularly interspaced. Repeat, repeat, repeat with these spaces in between. Short because they're only like 20 to 30 base pairs long, each of these things. Palindromic because the repeats read the same way forwards as they do backwards. So there we go. This is CRISPR. This is what we're referring to. And here I've only got two spaces, which means this one has actually come from this virus, but this one will have come from another virus. And as I said earlier, some bacteria...
Okay, so you get that? The spacer, like this whole thing, can copy there's the protospacer from different virus. So let's say you have virus 1, the bacteria copied it here, and you have the second virus, it copied here, and probably the third virus, it copied it. It sort of keep the memory within the bacteria's genome. Yeah.
An archaea can have hundreds of these CRISPR, hundreds of these spaces, so very long CRISPR arrays. So the question is now, what happens? Well, this is double-stranded DNA. Bacteria have circular double-stranded DNA. And what we need to do is we need an RNA polymerase to come into action.
All right. So what is the function of RNA polymerase is, okay, remember about the first document that I mentioned about DNA, okay, and then transcribe into RNA, messenger RNA, before the messenger RNA can do the next process, which is protein production to form protein in our body. So RNA polymerase, the function is to transcribe, yeah, transcription of the DNA here to RNA. Okay, let's look at the rest.
An RNA polymerase. What do RNA polymerases do? They turn this DNA sequence, transcribe it into RNA. Now, in humans, we'd usually transcribe this into what we call messenger RNA, which is a message to be able to transcribe amino acids and therefore proteins. But that's not what's happening here. So this is not called messenger RNA. This is actually called pre-CRISPR RNA.
Okay, so now it is different with human. RNA polymerase in bacteria is used for transcription of this CRISPR array DNA into RNA, and it's called what is called that? Pre-CRISPR RNA.
So now we've got pre-CRISPR RNA, and as you can see, it is a stretch of repeats and spaces. Wonderful. That's what the RNA polymerase has done. Now, another RNA polymerase will go to another discrete area of the bacterial genome and actually transcribe other types of RNA. These RNAs that it actually creates or transcribes, parts of them match the repeats. Sorry, these parts here. So parts of it will match the repeats. So if we were to draw it up, what we're going to have are these different sequences of RNA where parts of them match perfectly with the repeats. and other parts don't.
Okay, so if you listen to him just now, RNA polymerase also transcribe a different, probably DNA, into what they call, what is it?
And
The
so we can put
name
the
is,
binding areas in like that, and then other parts won't bind, but then other parts will bind, and then other parts won't.
it is
Now,
called,
what these strands of RNA are called, they're called tracer.
okay. So let me repeat. So the RNA polymerase transcribe, do the transcription process of this CRISPR array to one to pre-CRISPR. Okay, all right, and also it transcribe to what we call as trace CRISPR. Okay, and what's the difference is this DNA, sorry, this tracer RNA, okay, this one is pre-CRISPR RNA. This tracer RNA, it has certain match with the repeat, the red one, but certain part does not match. So that's why it has this like curve, some straight, because this one matches, this doesn't match. So these two are transcribed by RNA polymerase from the DNA of the bacteria.
RNA. These are called tracer, T-R-A-C-R, tracer RNA. So what we've now transcribed is two RNA strands where we've got a pre-CRISPR RNA and then a tracer RNA that's bound to a repeat, but we've got multiple tracer RNAs. This is where we need to bring in an RNase. So it's an enzyme that breaks up.
Okay, so there's another enzyme involved, RNase. Okay, this is RNA polymerase. Okay, usually we can imagine in chemistry we have polymerization, so this helps us to make it easy. It polymerize. Okay, and there's another one enzyme involved, RNase. Okay,
RNA. So what the RNase does is it comes along. So this is the RNase. It comes along and it will chop the spacer and it will chop part of the repeat. Like that. And it will do it at multiple areas. Now let's just do this one here, right? So we're using this particular spacer. So we've got the entire spacer, part of the repeat, and what it ends up looking like now is like this, where we've got the spacer,
repeats,
part of the repeat, and the tracer, which is bound to part of the repeat, and then we've got like a tracer tail.
The
like
tracer
that. So
here,
we've actually got two separate types of RNA. We've got now, it's not called the pre-CRISPR RNA, we've got the CRISPR RNA. So we've now got CRISPR RNA, and we've got trace. Okay,
so remember, just to recap back, okay, so we have RNase enzyme that cuts the spacer, the blue color, and bit of the repeats. The repeats is the sequence from the bacteria itself. Okay, and you have this, and it is combined with, because we have this bit of repeats here, and then this repeat matches the tracer RNA, some part of it. See, this comes from here. So you have this combination called CRISPR RNA. Okay, just now this one is pre-CRISPR, this one is tracer RNA. Combination of these two, we have CRISPR RNA.
RNA. And what comes along, the third thing we need is a protein, and this is called Cas9. So what Cas9 does is it's a protein that binds to these two RNAs. It binds like this, where you've got the spacer sticking out, but embedded inside, you've got part of the repeat and you've got the tracer. Now, your question might be, why have we transcribed these tracer RNAs? We've transcribed them because the tracer holds onto the Cas9. That's what it does. It allows for this RNA to be able to bind into it. So these are the three things we now have. Now, in actual fact, once the CRISPR RNA and the tracer RNA is inside of the Cas9, we can call it guide RNA. So we can actually...
Okay, so now we have a combination of CRISPR RNA, tracer RNA, and Cas9. So the whole thing, it is called guide RNA. Okay, so look at your Microsoft Words, the Word file just now. Okay, take the guide RNA. Okay,
call these two guide RNA together once it's in the Cas9, gRNA. Now, in actual fact, if we were to look at this in a little bit more detail, it would look a little bit like this. So let's have the Cas9 protein, and then we're going to have the spacer,
right,
like that, and then we're going to have the repeat up like that, and then we're going to have the tracer bound to the repeat with a little bit of a tracer tail, like that. So still two separate, there's one RNA and there's
okay, so this is the same thing actually. It's just that instead of horizontal, he made the repeats to be vertical. That's it. And this is the tracer RNA, right?
Another RNA. They're two separate. This is where we start thinking about gene editing and gene therapy. What they discovered, these researchers, was that you can actually connect the two together and you create a single guide RNA. So it's no longer a gRNA.
Okay, now instead of just gRNA, guide RNA, we can combine the repeat just now, okay, here with the tracer RNA, and this whole thing is known as single guided RNA, sgRNA. Sorry, why? Okay.
Right? We can call it an sgRNA, a single guide RNA. And that means we can actually make this whatever we want, as long as it binds into the Cas9. We can manufacture in the lab a single guide RNA that tells this CRISPR protein, or this Cas9 protein, I should say, where it needs to go in the genome.
Right. So this is actually the technology that is used in biosensor, because remember about the DNA probe, okay? And I mentioned about the DNA probe has to be designed to match the bacteria or the virus that you wish to sense. So same goes to CRISPR technology. We need to design the single guide RNA sequence so that it matches the bacteria or the virus that we wish to detect. And once we have designed this, we combine it with Cas enzyme. So Cas enzyme, there are different Cas, Cas9, Cas12, Cas13, which I cannot cover in this discussion, but enough that we need this for the development of biosensor. So similar to DNA biosensor, the DNA probe has to be designed. The single guided RNA sequence has to be designed before we can combine with the Cas9. So let's look at what is the function of this single guided RNA.
Because what its function is this. When we get invaded, let's just say, by another virus, and that virus incorporates its DNA. So let's say we've now got double-stranded DNA being injected by this virus. What this now does, the Cas9, is it comes along, it has its spacer, and the spacer will be complementary to one of the strands of the virus, remember? And then we're going to have the part of the
repeat
CRISPR RNA, or part of the repeat, and then we're going to have the tracer. And the point of what's happening here is once this guide RNA is bound to this viral RNA that's now been injected, it identifies where it needs to cut. It goes, okay, I'm bound here, right at the end, I'm going to chop it up right there, and I'm going to chop it up right here on the other strand. It breaks both strands. Cas9, chop, chop. And now what we're left with,
okay, remember this process. So look at this, look at here. This is the process of the editing. Yeah. So this line, this gene sequence or nucleotide bases, sorry, comes from the spacer. And this one comes from the repeat here. And this is tracer RNA. And the whole thing is called single guided RNA together with the cast. And then when the virus reinfected the bacteria, now this whole system will recognize the virus based on this spacer sequence. So it sort of, once it is recognized, you can see that they know because of the genes of the memory is copied here. So now they can recognize the gene sequence. So the whole single guided RNA can start to edit or chop these certain parts, yeah, here and here, okay? And that's how the editing can be done.
Is this double-stranded break in the DNA. So we've now got DNA that's got this
broken
break. Like that. Now, what does that mean? This is viral DNA. This virus has invaded, but we've chopped it. Viruses don't contain their own enzymes to be able to fix this break. So that means the viral RNA or DNA has now been broken and it cannot be fixed. Brilliant. We've now inactivated the invading virus. What does this mean for gene therapy and gene editing? It's all about, if this was in a human being, think about that. If we utilize this CRISPR technology with this Cas9 and the single guide RNA, and we chopped a very specific part of our DNA like this, what does that now mean? Well, think about it. Humans actually contain 2 copies of every gene. So we've actually got another gene down here.
So this is the, if we use it for gene editing in humans.
Now, if we want to repair this break, what can happen is 2 different types of repair mechanisms. You can have what's called non-homologous.
Okay, so this is where I stop, because we're not talking about gene editing in human, but the process of how single guided RNA is produced, because this is our biomarker. So this is the most important for us to focus, because this is the one that we want to use in the development of sensor, okay, biosensor. Okay, so for the next 8 to 9 session, I already put it in the Putra Blast, okay, but I will show which minute that you have to look at. But these are the slides that is provided. So I hope that by now you are familiar with the terms spacer, repeats. Okay, before spacer is protospacer, and then you have PAM. Yeah, PAM is how the cast at the beginning, where to cut the protospacer. Okay, they identify where they need to cut the protospacer. So this is the PAM. This is the protospacer. So this is the first cast. The first cast trying to cut the protospacer before it is copied into the bacteria genome. Okay, so this is PAM, sort of indicator where to cut. Okay, so this is where PAM comes in to just to show the cast. This is where you cut. Yeah, and then I also have shown you the transcription of pre-CRISPR, and then we have tracer RNA, okay, in the process. And also we have guided RNA, and in the end, combination of CRISPR, tracer, and Cas enzyme, we have the single guided RNA, right? So which is important for us to use in the biosensor development. So once we have that biomarker, so the rest of the thing is what we have learned in the sensor technology. So it's just embedding um, the biosensor, the biomarker into the sensor system. Okay, so you can use electrochemical method, you can use optical method once the biomarker is done. So the process is getting the biomarker done. So that's it. Yeah. So I think in the slide as well, you can see that this is one of the sensor that has been developed by Broad Institute. Remember that I mentioned just now, Broad Institute is a combination of experts from MIT, from Harvard. Okay. So this is how it looks. This is the Cas enzyme combination with uh, single guided RNA. This is the whole thing is called single guided RNA. And then when it identify the sequence that is similar, so they can cut. Okay. So remember the identification in biosensor is similar, like we use DNA probe to identify the specific gene in bacteria. So this is the same things, okay? Identification of a series of gene sequence in the bacteria or in the virus, but the mechanism is once it is identified, it cuts, it edits, okay? So the cutting process is used to sort of transduce, yeah? Being transduced by the transducer, and we can read the signal. So remember about our basic understanding in a biosensor or chemical sensor. So if you want to read more, you can have this a lot of papers on the uh, development of biosensors using CRISPR. Okay, how it works. So, right. This is what happened. The word cleavage here is the cutting process. Yeah. So you can see with the cutting process, it is transduced into increasing or decreasing of current. Yeah. So that one is something that we have learned before. Okay. The redox indicator, right? So what I have presented or discussed with you just now is the principle of how the biomarker based on CRISPR technology is developed. Once the biomarker is prepared, then we can use the biomarker or the bioreceptor to be in the electrochemical base or optical base sensor. So this is what I would like to share with you today. So the next one that you probably can watch is this one. I have shared with you the lecture slide and the CRISPR supporting just now, the word, all the terms that you need to know. And for this one, there are there are other technique being discussed. It is called LAM. So if you want to skip it, just straight away go to um, uh, just a moment, not this one. You can straight away go to uh, because I think there's something wrong with the
different type of Crispr.
Okay. It's supposed to start at minute.
Last week, they tested.
It's supposed to start at minute 30 minutes. Okay. But unfortunately, there's some error. So
we are in a century that you're not only subject to one area. For instance, since I am
okay. So the best is you view the video at 1 hour 10 minutes. Okay. So this is an invited talk by Professor Mehmet from Turkey. Okay. So you probably have difficulty listening to him. But try your best since I have discussed with you on the terms, so at least you understand the terms. But he explained on the application of this CRISPR technology in biosensors. So please do listen to him. And you can also repeat back uh, what I have no, what I have discussed just now from this link, all right, in YouTube. So I think that's it. Okay, let me go back to the okay. I think that's it.